to episode 65 of the Breaking Bio podcast. I'm Stephen Hamlin, postdoc at the University of Southern California now. Let's say University of New South Wales. And we're joined here today by Heidi.
Hi, I'm Heidi Parker. I'm a postdoc at the University of Texas at Austin.
And special guest today, Melissa Wilson Sayres.
Hi, howdy. I'm a postdoc at UC Berkeley right now.
So, Melissa, for the audience at home, can you tell us a little bit about what you do?
Yeah, I was going to start. So as an undergraduate, I was a math major, and I applied to PhD programs in mathematics and in bioinformatics, and I decided to go the bioinformatics route, which meant that when I went to graduate school, I wasn't entirely sure what I wanted to do. So a lot of people have a good idea of the kind of research area they're interested in. I did not. I did 3 rotations. One of them was working with yeast genomics and doing chromatin immunoprecipitation and microarrays in yeast. And then the second rotation was working with Arabidopsis and microRNA. And the third rotation was working on sex chromosome evolution. And that's the one that really got me. And I was like, this is what I need to do. And it was staying up asking questions and reading papers. And so I found out exactly what I wanted to do. And so what I do now is I study sex chromosome evolution. That could be any chromosomes that spend different amounts of time in the male and female germline. Primarily, I study the X and Y chromosomes in mammals. The X chromosome has about 1,100 genes on it. The Y chromosome, some work I've done has shown it's lost about 90% of the gene content that it used to share with the X chromosome. And so trying to understand those dynamics, what's important about what's retained on the Y chromosome, do we think the human Y chromosome is going to be continued to degrade over time? And what can we learn about dynamics of human populations by studying the sex chromosomes versus the autosomes?
Right, so I actually just read one of your papers a while ago, I think. It was the one in PLOS Genetics about Y chromosomes.
Mm-hmm.
Or at least I saw it. So can you tell us a little bit about what the findings from that were? Because I found it interesting at the time.
I'm glad you did. So the recent PLOS Genetics paper was looking at diversity in human populations on the autosomes, which are non-sex chromosomes. The X chromosome, genetic females have 2 X chromosomes, and genetic males have an X and a Y. And diversity on the Y chromosome and diversity on the mitochondrial DNA, which is a small circular bit of DNA that's inherited through the genetic maternal lineage. And what we found when we were looking at diversity, just variation across populations, was that the variation on the Y chromosome was much lower than we expected. And this could be for 2 main reasons. And they're not necessarily in conflict, but there have been a lot of papers on either side of this. And one of them said that one side is that there's low diversity on the Y chromosome because there's fewer males that are contributing to the next generation. Just that there's higher variance in male reproductive success. So this means some males father many children and some XY individuals father very few children. And so we did some modeling, statistical modeling, and we said, what are the expected levels of diversity, the expected variation on the autosomes, the X, the Y, and the mitochondria at different ratios of the number of males and females that are contributing. And even at a very low male to female ratio, that wasn't sufficient to explain the low Y diversity we had. And so we did some more modeling, and the modeling showed that if natural selection is acting on the Y chromosome, so even this chromosome that has 27 unique genes, it's less than 1% of our genome. If natural selection is acting on it, that's consistent. That allows us to find a model that's consistent with the low diversity that's on the Y chromosome. So that's really exciting. It means that it's not probably just going to wither away into nothing, that the genes that are there are still important in humans anyway. It might be different in different species. There are some species of mammals that have lost their Y chromosomes. There's an Okinawa spiny rat that females have 2 X's and males just have a single X. They're called X-naught. They don't have something else there. And what we think happened is that those core set of really important Y-linked genes got moved to one of the non-sex chromosomes, and they're going to have a brand new set of sex chromosomes. We just haven't really identified them yet. So there's a lot of ways you can have chromosomal genetic sex determination without very distinctly different X and Y chromosomes. But humans, we just have really old sex chromosomes. So our X and Y's look very different.
Hmm.
In the Okinawa spiny rat, do you see like what kind of mating system do they have? Is the male to female ratio 1-to-1?
I don't know that that's been well studied. So there's a lot of islands in a lot of Okinawan islands here, and it's a little flippant to say that it's just this one Okinawan spiny rat that has lost its Y chromosome, because there's actually a lot of different species, and each of these island populations has its own unique weird sex chromosome system. So some of them have their XXY, some of them are X naught. In males, there's a lot of different weird, unique things that are going on, and we haven't studied them very well yet. So.
That's amazing.
So some of our audience is pretty varied, and some of them aren't going to be all that up on population genetics or sex chromosomes for that matter. So can you break it down for a wider audience what the implication of your results are for humans and sex chromosomes in general?
Well, I think one thing that's important is that the Y chromosome, there's a lot of things that are unique about it in our genome. All of our other chromosomes, in humans we have chromosome number 1 through chromosome number 22. You have one copy of that inherited from your genetic mother and one copy from your genetic father, and they look almost the same. There's some teeny tiny differences, but very broadly they can bind and swap DNA. If my arms are chromosomes, they can swap DNA over their whole lengths. The problem, or not the problem, but the difference with the Y chromosomes, there's this big X chromosome and this teeny tiny Y. Maybe it's just like that. And so it can bind up here. It can pair in a tiny pseudoautosomal region, but for the most part, the Y chromosome doesn't have a partner to swap with at all. So it doesn't swap DNA. There's no recombination on the Y chromosome. It leads to it has a much smaller effective population size, which just means there's fewer Y chromosomes in the whole population. If you just imagine two people, a genetic male and a genetic female, between them, they will have 4 copies of the non-sex chromosomes. The genetic female will have 2 X's, and the genetic male will have 1 X. So there'll be 3 X's for every 4 non-sex chromosomes. But there'll only be 1 Y chromosome for every 4 non-sex chromosomes. So that means, one, that you're just going to have fewer Y chromosomes going around in the population. So one thing that is good about that is you can use it to trace populations. So it's smaller, and there's only 1 copy, and it only goes through the genetic male lineage, so you can identify, well, who was that genetic male's father, who was that genetic male's grandfather and great-grandfather, and on up and on up. What our stuff shows is that tracing back is probably fine, but maybe something that geneticists do is they assume a constant mutation rate over the tree. So they say that, okay, with each one of those generations, you can imagine it like a ruler, and you could measure, okay, if we know how very different all of these are, we can measure exactly how long ago these Y chromosomes split from each other. But if natural selection is acting on the Y chromosome, it takes the ruler and it squashes it down. But we don't exactly know how much it squashed it down. So what we think we're measuring as a centimeter, maybe it's only a couple millimeters, or maybe it's 5 millimeters. But we don't have a really good sense of that if we haven't looked at the amount of natural selection that's acting there. So I think one of the main implications that we need to study that a little better before we really trust those absolute measurements, right? If we say, like, all something they do, Ashkenazi Jewish individuals, if we can trace all those Y chromosomes back and we say that we find that Y chromosome, the most recent common ancestor of all those Y chromosomes was however many thousands of years ago, 30,000 years ago, or 10,000, 3,000, however many it is, our ruler isn't right. The ruler that we're using isn't right, and so we need to work on that.
Oh, sorry.
Go ahead, Mike.
After you'd done this study, did you feel like, wow, men are almost useless? Or did you feel like, you know what, there is still some use, it's not totally worthless?
So one of the Y chromosome is not synonymous with being male. And the Y chromosome is found only in the genetic male lineage, but there are people with a Y chromosome that present as female. And that's not even gender identity. There's a whole spectrum of intersex individuals. And so when I'm studying X and Y chromosomes, although I'm steeped in this gendered language of male and female, I tend not to think about it that way. I think about this Y chromosome that's passing through the genetically paternal lineage, but that I don't often think of these conflicts between males and females. And moreover, the Y chromosome is only 1% of the genome. Most of our genes that are expressed in a biased manner towards genetic males or genetic females are on the non-sex chromosomes. We just haven't identified all of them very well. In fact, most of them are on the non-sex chromosomes. And so while the sex chromosomes are a good place to start, they're a good place to really put our magnifying glass first for looking at sex-biased patterns. It's not the only place, which is kind of cool, but we need to do a lot more work before we can use the rest of the genome.
Yeah, I saw there was an androgen receptor that's expressed on one of the sex chromosomes in frogs, in a species of frogs, because they're very different. You know.
Yeah, I think the cool thing so there's a suite of genes, some set of genes that are involved in sex determination in mammals and in birds and in frogs and in fish, and a good proportion of that set overlaps across all these different species. So even though in some frogs they have X and Y, or Z and W, they have different sex chromosomes, it seems like often the genes that get picked up to be involved in sex determination are very similar. They're androgen receptors, they're transcription factors. Their transcription factors turn on and off different parts of our genome. And I think it's super cool that there's this set of genes that, regardless of what species you are, we seem to evolution keeps pulling at those genes over and over. Maybe it's not the same one. So the sex-determining region of the Y, this switch in humans, it's not the same as it is in birds or in frogs or turtles. Something that's downstream of it is actually upstream in another species. So all of those things kind of work together. I think that's really fascinating.
So the upshot is that the doom and gloom moral panic that erupted a few years ago about the Y chromosome going away and males being eliminated from the population, probably not going to be a huge issue after all.
Yeah, that was one of my papers too. But yeah, so the Y chromosome may go away. I'm not worried about that. Males will still stick around. Unless we become like, there's some birds and lizards undergo parthenogenesis, where in the females, at the last division of the egg, where you just have half of the genome content, so you get half from your mom, half from your dad. In these lizards, what happens is that it starts to separate, and then it's like, no, we'll go back together, and they form clones of themselves. So we could do that. Probably.
That would be amazing. That would be amazing.
Humans probably won't, but it's possible, and it's happened in some species. So,
if we did lose the Y, how would the functional aspects of like SRY and such be I don't know, adapted, taken care of otherwise?
It probably, so it's a cascade pathway. So either SRY, so one option is SRY, sex determining region of the Y, would jump to a different non-sex chromosome, and that non-sex chromosome and its partner would become a neo X and Y. That's one option. Another option is that a different gene would become the sex-determining switch. Instead of being SRY, there would be a different gene in that pathway that would become the switch. And both of those have happened in mammals and in other species.
All right.
So, in the future, oh, sorry, I just keep interrupting you. Go ahead, Stephen.
No, go ahead. Because I'm going to switch topics, so you go ahead.
Okay, I was going to ask, like, you've studied in humans and you're mainly focused in mammals. Are there the systems that you want to stay in?
Oh, I'm very happy to study any system. I think there's a lot that we still don't understand in mammals, and especially humans. A lot that has to do with intersexuality. That's not very well studied, which isn't specifically the X and Y chromosome. That's across our whole genome. I think that's fascinating. But really, if it has separate sexes, I'm interested in it. So.
There's a lot of stuff I want to touch on, including like your science outreach and living as a scientist. But I'm going to throw one to you out of left field here. Recently, well, I guess about a year ago now, E.O. Wilson wrote a famous article in which he suggested that mathematics not really a big deal for biologists. And I'm wondering, as somebody with a mathematical background, which we don't often have on the podcast, how you feel about it.
I wrote a blog post about it. I was so upset about it. I'll send it to you.
To the internets.
Yes, yeah. I was thinking if I could find it real quick, but I'll focus here.
We can put it up in the show notes.
Yeah.
Yeah, I think so. Initially, how that article came off to me was, I'm an old successful person, and I didn't need math to be successful in biology, so you don't either. And that seems like a poor way to give advice. I think if we always think about what was successful in the past, then we're never going to make a lot of progress in the future. And the way that technology is advancing, does everyone need to know differential equations? No. Does everyone need to know multiple regression or pick whatever part of math or statistics you're particularly interested in? Everyone does not need to know modern analysis, but people need to not be afraid of it, and they need to realize that the tools that they're using are not black boxes, or they should not be black boxes. And even if you can't write down the particular formula, you should be able to understand the basics behind it. Having collaborators is great, but you need to understand what your collaborators are doing. So even if you're doing experiments and you have someone who's doing field work and someone who's doing the molecular biology aspect of it, whoever's the molecular biologist should know what's going on in the field, right? They're not going to be going out and particularly catching the frogs and taking their toenail clippings, but they should understand what that process is like. And likewise, whoever's in the field should understand what a Western blot is, or what mass spec is, or whatever the particular feature they're doing. And similarly with mathematics, I think you still need to have a good idea of what's going on. Now, that doesn't mean that it's not scary, right? Math is scary to a lot of people, more so than other things. And part of that is how we teach mathematics. And there's a lot of people that are getting better at explaining it. There's a lot of free resources for learning more about math, but I guess one of my aims in talking to people and doing outreach is to try to get them to be less afraid of understanding the broad concept of the math or the statistics that's going on in their projects, even if they're not actually going to be doing it.
It's a shame Morgan isn't here because he's terribly afraid of math. So
I would say I'm not afraid of math, but I didn't take that. So Ed Wilson also wrote a book called Advice for Young Scientists, and he talks about issues like this. And I take it more of collaborate with other people, know what you know and know it well, but you can always collaborate. There's a lot of computational stuff that I don't know, and I know enough to know that it's part of my paper that I would never put my name on something that I didn't understand. But I don't know if I could do it on my own without, my collaborators sitting there helping me with it.
I find that's an odd tension. Sorry.
Go ahead.
I was just going to say, I find an odd tension in that in biology, especially, where there's a lot of people who they fetishize stats and how to do various, they'll know all the intricacies of linear mixed models, but you ask them to do something with a differential equation and they run away screaming in terror, and I've never really understood why. I don't know.
I think it's something we don't practice that much, but we like to say that people who like differential equations, like you, Stephen, don't know anything about animals.
Yeah, I think part of my response also was that I had friends who said, I just need to put this equation into R, or put it into whatever, or put my data into it, and out will come the answer. But there's been survey studies that have shown, just absurdly ridiculous amounts of p-values that are reported are improperly applied tests, or they're improperly rounded. And it's so frustrating that we focus so much. There's this huge spike of p-values that are 0.049 or something. And
I can do one better than that. There was an undergrad who my friend's TA for that had a p-value of 8.
Yeah. So, for example, so people should understand if I get a p-value of 8, that's
right.
Something has gone wrong somewhere in the pipeline, and if you try to publish that, then there's something worse. And so I feel like maybe in the book he went into more detail, but in that one particular article he had online, it wasn't so clear. It was like, it's okay to be afraid of it, and it's okay to not have to study it. And I took issue with that.
You know what I think would make people less afraid of math is that, when you were younger, were you on the math team? Anybody?
Yeah.
Yeah. Yeah, so we had t-shirts, we were mathletes, and it was so much fun. And you would go, and the individual tests were fine, but the group test you would take with your friends and working down these like massive problems, I love that. I would do that now. If they had that in place of like pub quiz night, I would be all over it.
With the exception that was pretty much social suicide.
I mean, I went to a small school.
I did too. Yay.
Yeah, it was totally fine. I did it in high school too.
I did not, and you got beaten up for being on the math team, like I did.
I don't think anyone ever knew.
Yeah, nobody would care, right?
Yeah, it was like...
There's a lot of people working on math education and bioinformatics outreach education, which is slightly different than working with scientists who are professional scientists. And in a way, it's almost easier to go do high school outreach than it is to try to convince PhD scientists that they need to learn something new.
Yeah, I could see that.
So that's actually a good reason to segue here, because you actually are involved in a lot of outreach, aren't you?
I try to, yeah.
So tell us a little bit about what types of outreach you like to do.
So in graduate school, I did a lot more than I have as a postdoc. I designed a biannual workshop for Girl Scouts to work on science badges, and so we had graduate students, for each one it would focus on whatever the area was. And it's because we talked to some parents of these Girl Scouts who were in middle school, roughly, and the parents said, oh, we feel pretty comfortable doing all of these other badges, but the science badges, we don't feel comfortable teaching them what they need to know. We don't want to teach them something wrong, so we just don't do it. We don't teach them and let them do their science badges. And so something at Penn State we started doing was once a semester, organizing an activity. We'd have a day-long workshop. Girl Scouts from across the state of Pennsylvania, I was at Penn State University at the time, would come in and we would do these fun things. So sometimes we would do archaeology, we did oceanography, we did an engineering one. So those were all pretty fun. I did some molecular biology outreach at a high school. Every year we would go in and teach some classes, teaching them about gel electrophoresis and DNA extraction. So we'd extract DNA in these little tubes and have the students talk about that and answer questions. Since I've been a postdoc, I've done outreach at the local high school just teaching phylogenetics. So I go in and do a lesson, like what is a phylogenetic tree, what can we learn from it? And more recently I've started doing some outreach more with adults. So I've given a couple public lectures, and I've gone to this conference on world affairs, which is this huge conference on everything conceivable. And we go and sit in on a series of panels and talk about different science that's going on, and then talk with people in the intermediate about my own research. So I think all of those are really valuable for showing scientists as real people and for talking about our experiences. So on one panel, for example, I just talked about the process of becoming an assistant professor, and people were flabbergasted that you do undergraduate and then you go and you do between 4 and 7 years of a PhD, and that's not sufficient at most places to get you a tenure track position. So then you do a postdoc, at least one postdoc, and then you might do another postdoc. And they were just staring at me, why are you still in school? And yeah, so I mean, this year I've been on the job market, and it's been especially present in my life. But I think it's easy for scientists to seem like they're part of this ivory tower, right? We don't talk to people a lot, maybe because we don't know how to, or maybe we don't think it's worth our time. But to me, I think it's really valuable. How do we get science funding? We get science funding because legislators vote on it, and if they don't know who we are and what we do, and they don't know because their constituents aren't telling them that science is important, then it's kind of this whole feedback loop. I need to be involved in that somehow.
I've kind of wondered about that myself. I don't know how you sell science as a career. Like, what you're saying, yeah, you've been this many years in school. Most of the people have gone on and they've done all the life plan, boxes, and you're still studying. And I know it's because we do it because we can't imagine doing anything else. But how do you actually go to a high schooler and say, look, if you want to be a scientist, this is how you do it, and not watch them run away in terror? Like,
well, I think we don't need to. So here's the challenge: we don't need to convince everyone to become a PhD scientist. There's already a lot of PhD scientists, and I don't think that we're going to have a deficit of PhD scientists anytime soon. Definitely, we need to work on different demographics that are underrepresented in science. But when I think of science outreach, it's more about just general understanding of science and specifically not being afraid of scientific results, trying to understand that uncertainty is not the same thing as being unsure. That we can have some level of uncertainty, and that's actually a good thing. As a scientist, I'm more distrustful of someone who's absolutely certain and doesn't know what problems with their data are than with someone who can explain what are the problems with it. But that's completely the opposite of working with the general public, and especially with the court systems, right? If you talk about uncertainty, then people say, like, oh, well, so you don't really trust it? No. We do, but there's this little bit, this confidence interval that we have that tells us how much we trust it. So.
Yeah, I think overall improving scientific literacy is really important, and that would help with a lot of climate change and other issues, that people would understand that this isn't a debate. This is the issue that's happening. The world is literally ending.
Well, one of the panels I sat on, I thought it was really, they put me on a panel at this conference on controversies in science. And as an evolutionary biologist, in my mind, I thought, oh, you all want me to talk about evolution and creationism, right?
That's not a controversy, right?
Well, and so I started by saying it's not a controversy, but there is a controversy among scientists about this. And the controversy is, how do we as scientists deal with creationists? Do we engage them in debate, or do we just ignore them? Or some third option, but primarily, should we or should we not engage them in debate? And that is a controversy. I think people don't agree in science what's the best way to deal with that. I mean, I have my own opinion, but I think when I was talking about it, I framed it that way. Yeah, scientists, yeah, evolution happens. We're good. But how do you deal with the public? And we're not trained to. So it can be difficult, right? We're not trained to be in a debate. But it also can seem immensely unsatisfying to have someone spewing nonsense and not respond to them.
Have you run into that in your outreach efforts? I mean, you've been at the high school level.
Yeah, not at the high school. Not. I've worked with first graders. We did dragon genetics with first graders, middle schoolers, high schoolers. I only once have I had someone, and it was an adult, come to me and ask well, I mean, it wasn't really a question. It was kind of a leading. Have you read Bible? And I was like, yeah, I have. But it was really it. I'm surprised it hasn't been more.
Yeah, because I tend to interact with a lot of these people on Twitter, and I'm just running into a wall constantly.
It's a lot of energy to deal with those sorts of things, and there are a few people, like Josh Rosenau is one of the people who, I talked to him once, and he has this whole website talking about what organizations, including churches in North America, that do not advocate the teaching of creationism in school. So sometimes when people get crazy religious, I refer them to these sites because it's just a lot of frustration. I'm already doing a lot of frustrating experiments, I try to cut down.
Yeah, I think a lot with this, just the evolution and creationism, most of the public, it's like watching your parents fight, right? Like, you don't want to be involved. If there's people that are arguing all the time and you don't really know what's going on, so you're just like, all right, you guys talk about it. I'm just not going to think about what's going on. I don't want to listen to people arguing. And so it's nice to have a site that says like, oh, yeah, we don't really disagree. Here's a bunch of things. Here's a bunch of organizations. I like that very much.
Yeah. I feel so bad for Bill Nye. Like, can we send him on a cruise every year? Why is he like the only guy that ever represents everyone else? Damn.
Well, people were really upset. Some people were very upset at him because he's not an evolutionary biologist, and he was completely unprepared for that. And he could have been. He could have been, because there's the National Center for Science Education, right? I mean, there's a ton of information there. Someone there could have been really good at.
You could have asked anyone. Like, I don't think Bill Nye has a PhD. Not that I don't love Bill Nye the Science Guy, right?
Yeah.
He's no Mr. Wizard. I don't know if you guys have Mr. Wizard, but
Yeah.
I don't know what Mr. There's some kind of fix-it shop down the road that's like Mr. Wizards, and I always wonder, like, oh, this is where he retired.
Oh, that's awesome.
That would be amazing. He's just like there doing dry ice stuff every day. A lot of dry ice on that show. All right, so we're actually running out of time.
Yeah. So,
is there anything else you want to put in?
Oh, I don't know.
thanks.
Finding an academic job is hard.
You know, there was a talk here yesterday. They have a lot of postdoc talks, and there was a Brown Bag session on how to, like, forge your own path in science and sort of, like, distinguishing yourself during your PhD and your postdoc. But do you apply to EB departments, evolutionary biology, but also, like, medical schools? Because what you do translates to humans.
Yes, yeah. I applied to medical schools and ecology and evolutionary biology and general biology departments, and I'm a better fit for an ecology and evolutionary biology department, but it's difficult because they're very different. The focus is very different in those different departments. So yeah, it was really interesting. I think I went on 12 interviews, so very different departments. So I also interviewed at a liberal arts college, a primarily undergraduate institution, and R1 institutions, and second-tier institutions. So I had kind of all of those in the line, and maybe that's partly because of the kind of outreach and stuff that I did. I wasn't specifically focused just on research, although that's my primary goal, is to do that. I want to be somewhere where I can still do outreach.
Yeah, that would be great.
Yeah. We'll see. Hopefully in a week I'll know.
Yeah. Well, we'll have to have you back on to talk about how that turned out. There's actually
we could have
so
a whole
much.
half hour on jobs. That would be easy.
There is so much that we'd like to talk to you about, but we are actually out of time at this moment, so we're going to have to wrap it up here.
Yeah.
How can people follow along with your ongoing travails on the internet?
So on Twitter at @MWilsonSayres, or I haven't been blogging as I want, but I'll get back to it. MathBioNerd. So .blogspot.com. So both of those, and I'm pretty open on social media. So if people ask me something, I'm happy to answer it.
All right.
Awesome.
So if you want to get a hold of us, we're at Breaking Bio on Twitter, and you can email us at breakingbio@gmail.com. I'd like to thank Melissa for joining us for episode 65, and we'll see you guys next time.
Thank you.
Why don't you tell us a little bit about what you do for the audience?
Okay, I'll start a little bit further back. So I was an undergraduate.
What the bloody hell? I'm sorry.
What? No. How is this happening? Stephen, why are we recording at the Gong Show?
Oh, no.
Why have we been gonged? I'm sorry, Melissa. I didn't gong you.
Son of a... I have to be outside at the moment, and so, I don't know, somewhere there's like an air raid siren going off. Are you kidding me?